Setting up your meeting.
I'm gonna just share this on Instagram that we're live.
Oh, that would be great.
Yeah, if you guys want to just talk for a second and
Setting up
introduce
your
us or
meeting.
something. I'm gonna
Yeah,
just share this on Instagram that we're live.
Oh, that would
if
be
it's
great.
live, we can,
If you guys want to just talk for a second and
Setting up
introduce
your
us or
meeting.
something.
yeah,
A little bit of feedback.
How's that?
yeah,
Is that
there's a delay going to YouTube when you connect it.
we are live now. So, okay, let's get going. Thank you
Right.
everybody for joining today. I'm Alyssa. I am on the technical side of things for Pikes Peak Mycological Society. We are here today with Mycota Lab, Stephen and Mandie. I think a lot of you guys know Mandie. She's done a lot of things for us. So we're happy to have her back. Welcome back. And they have a really fascinating webinar for us today on DNA sequencing. And I don't know much about it, so I'm just going to shut up and let them take over.
I'm just trying to share this on Instagram really quick, Steve, if you wanna I'm gonna share my screen. I have this nice PowerPoint, and then I'll let Steve talk for 1 second while I share this. Um,
Yeah, sure. So my name's Steve Russell, director of Mycota Lab, and we are currently working towards documenting all the macro fungi of North America. And so one of the key projects that we've been working on over the last number of years, I think we started in 2023 now, is something we've been referring to as the Mycomap network. And so we've been gradually expanding the number of areas in North America that can submit specimens, as many specimens that they would like, as they're willing to collect, dry out, and mail in. And they can submit those specimens for free DNA sequencing. And this year, we're actually opening up the entire Rocky Mountain region. So everywhere from Colorado to Wyoming, up into Montana and Idaho. And so it's a significant expansion. The region would really benefit from learning more about the macrofungal diversity that exists there. I guess I could give you a little more history about kind of where all of this came from. So back in, well, it would have been 2016, 2017, somewhere in that time frame when we started the North American Microflora Project, which is now known as Fundus. The structure of it for people was that there were individual projects that people could start and they could mail in specimens at that time for DNA sequencing. And we were able to make some progress there. One of the big limiting factors at that time was the cost of sequencing technology. And so back then we used a process called Sanger sequencing that was, $10 or $15 per specimen. And when you start thinking about the number of specimens that can be brought in by people, that adds up really, really quickly. So, if you're trying to do 1,000 or 2,000 specimens a year or something like that from a state like Colorado, that number, times 10 or $15 ends up being a big number really fast. And it generally exceeds the budget that's within most clubs' abilities. And so back then, early on, 2016, 2017, we started a project in Indiana that we really kind of wanted to try to make it be an exemplar project for what was possible, for what is possible in terms of documenting fungi for the state. And one of the key initiatives that we found to be really valuable back then was something that we referred to as online forays. And this is something that would be great for you guys in Colorado to think about as well as you start to submit more and more specimens. The general structure for an online foray, so if you think about a traditional foray, especially for a state like Colorado that's really diverse, it takes a long time to drive. If you hold a foray in one part of the state, there's going to be the majority of the state's not going to be able to make it over to it. There's a huge cost in gasoline, a huge cost in people's time. And so one of the biggest things that we found to be beneficial in our Indiana project is holding online forays. And so we actually do that. We've been doing that since 2017, twice a year. And anybody from anywhere in the state can just go out to their own lawn, wherever they are, their local state park, state forest, collect specimens and dry them out and mail them to us. And so that takes away a lot of the structural barriers that are encountered in traditional forays. And it's been one of the most beneficial things. And fortunately, I mentioned that it used to cost a whole lot of money to sequence individual specimens. And now it doesn't with new sequencing technology. We went from something on the order of $10 to $15 per specimen to maybe something around 25 cents per specimen. And that key feature has been really what's allowed our lab to scale up our sequencing efforts to incorporate other states, to include states like Colorado and the entire Rocky Mountains. Eventually, it'll be the entire continental United States. But that significant, 100x decrease in sequencing costs are really what has allowed us to even conceptually expand into these areas and to be able to offer this kind of service. And so, jumping back to Indiana for a second, and I don't know if Mandie mentions that at all in her slide presentation. I'll let her jump in here in a second. But we're probably north of 20,000 specimens that we've collected and sequenced to this point in Indiana. And so we actually have a really good understanding of the biodiversity that exists at these really large scales. And that's something we're hoping to be able to replicate in Colorado. We want to get as many collections as possible. We want to document as much of the fungal diversity that exists. And we hope to work with you guys as much as possible to be able to achieve that goal. So with that, I guess that's a little brief intro. Mandie, if you want to jump into the slides, I can jump in periodically as well.
Thank you,
Sure.
Steve.
Yeah, thanks for the great introduction. Yeah, so this has been, stuff has been going on since 2015, so it's about a decade. And let me see, okay, yeah. So our ultimate goal over the next decade is not just to have a really good grasp on Indiana or some of these other states like California, or Oregon, that have a lot of collections already, but we want to know what is the diversity in every state, really on the whole entire continent of North America. And Stephen really thinks that we can do that in the next decade. So yeah, I'm really excited to be part of this. So my job for my Mycota Lab is to basically do what I'm doing for you guys right now, but in California. So I'm the community science director in California. So I'm a hype man, basically. So I'm a hype man for people collecting mushrooms and sending them into my Mycota Lab. And so yeah, I've given this presentation several times to many people in California, but kind of made it just for you here in Colorado for this particular presentation. But the goal is really the same throughout the entire continent. So we want to document all the macrofungi in North America over the next decade. How are we going to do that? Well, we're going to create a strong network of community scientist collectors across the continent. And really, I think it'd be, we don't just think, but we know we've proven so far in Indiana and other states that a small group of dedicated people are really who can change the world. So instead of having 100 people sending in 10 specimens each, it'd be much better to have 5 or 10 people. But there's no real limit on it. If you guys want to do this, at least for the rest of this year, we're not going to stop you. We're here to support you. So funding priorities change, and like for instance, last year we didn't have an open call for Colorado, but Ohio Mushroom DNA Lab was accepting specimens from Colorado. Now, Ohio Mushroom DNA Lab is shut down and is not accepting anything. And you guys are just super lucky because Mycota Lab decided to open it up for this year. So who knows where we'll be next year? I would love if we could keep continuing this, but I really want to encourage everyone to strike while the iron is hot while we have an open call in this state. So yeah, and you guys really have an abbreviated season there in Colorado too. So it's like, is it even starting now, James?
Yeah, I've been collecting since April, actually. So it's not quite prime time, but we are starting to get stuff and we're getting a lot of moisture. So as soon as it warms up, we'll start to ramp up pretty good.
Yeah,
So
so
it's a good time to be doing this or talking about it, I think.
exactly. So yeah, so now through like September-ish, with the peak really coming after you guys get the heavier rains in July and August. Fingers crossed. Um, but yeah, it'd just be great to get as many of, people from this club doing this as possible. And this all goes through iNaturalist. So I know that people, I've given presentations for you guys, and a lot of people already know about iNaturalist, but iNaturalist is the bomb. It's amazing. It's the coolest and best social network. Um, you post a mushroom and people come on a lot of times from your local area and will help you with an identification. So this is the beginning of the workflow for all the mushrooms that are submitted for DNA sequencing. It all kind of flows through iNaturalist at the beginning and the end. So you'll put your mushrooms on iNaturalist, and then once your DNA sequence comes back, that will also be uploaded to iNaturalist. Yeah, so this is such a cool thing. This is, what I do for my job. So I realized this probably like 6 months ago. So if you look at all the INET observations in the US, there's 105 million, which is crazy. About 7 million of those, or 7%, are fungi. That actually tracks with the rest of the world. If you look at the INAT year in review, about at the end of every year, it's about 7% of all the INAT observations in the whole world are fungi. And then about 70,000 fungi on iNaturalist have DNA sequence data. So that's about 1%. That's in the US. About 1% of all the fungal observations have DNA sequence data, and that is absolutely amazing. I mean, no one can really say that anywhere else in the world about this particular kingdom of life. So we are really, really doing a great job. But if you look here, so we have about 70,000 barcodes on iNaturalist right now. But there's only, if you zoom into Colorado, there's only about that many fungal observations, 70,000, 84,000. It's about the same number. There's only about that number of iNaturalist observations in the entire state. And although this number has come up in recent years, it's actually doubled since this time last year, or it's almost tripled, honestly. It's like 2.5x, 627 is the number of fungi in your guys' state with DNA sequence data on iNat. So it's really, really small number, especially compared to other states. So like Steven said, there's 20,000 in Indiana. There's probably 12,000 or something like that, soon to be more than that in California. So, these other places are way ahead. And
Let me double click on one of the things that Mandie was saying, how a
yeah.
small group of people can be highly influential. So we've seen that in basically every state that we've opened up so far. If you look at Michigan, for an example, when we opened up Michigan, there was one individual in particular that ends up collecting somewhere between 2,000 and 3,000 specimens per year over the last couple of years. And so if you want to really get into it, if you want to really make a disproportionate sized impact on the biodiversity that's known in your state, it's well within the means of anybody to just decide to do it, to go out, to start collecting, to start organizing, to start drying, and to mail them in. And so I want to say it's well within the means of anybody who wants to do it, right? You just have to kind of make the decision. If you're going to do 2,000 or 3,000 collections in a year, that's going to be a part of your life, right? You're just going to have a constant flow of mushrooms coming in. And it's, essentially the decision to make a difference about your state's biodiversity. And it really does make a giant difference. But it's typically ends up being, a small number of people that make a really disproportionate impact. And anybody can make the personal decision to be one of those people.
Yeah, and I think a lot of people don't understand how easy it is to do it. And they don't understand that there are these opportunities to do it for free. And I think that's a lot of what we're here for, is to say, hey, we're doing it. You're doing it for Colorado this year. You know, usually I do the Myco Blitz, but that's about 10 specimens you're allowed to send every year. So this just kind of opens everything up. And since OMDL's not currently accepting anything, this is a great opportunity for everyone. And I think the big thing is just getting people to understand that they don't have to be, you know, a trained scientist to do this. All they have to do is have a cell phone, an iNaturalist app, take a picture, you know, collect some things, and then voucher it like so. This is one of our vouchers, but then I know Alan has this one that I've been using, which is really helpful. You can just put in your iNat number and it pulls all the information. So it's getting easier and easier to do it. And I think we just need to help people learn how to do it and show them how simple it really is, and just get the word out, really.
Right.
Yeah, I suspect Mandie is going to get into the protocols a little bit, but I can sum them up really easy, because we try to make the protocols as simple as upload to iNaturalist, dry out your specimens, mail them in. If you can do that, right? If you can go out, take pictures and upload them to iNaturalist, maintain organization of the specimens, dry them out, and write your iNat number on a bag, that's the bare minimum that we accept specimens with. And so, and there's lots of people that do that, that bare minimum, and that's perfectly okay. As you tend to get more into it, as you, you know, develop your camera skills, you develop your organizational structures, you know, there's other things you can do to help us out as well, but just the minimum requirements are as simple as taking photographs, drying it out, and mailing it in. And so it is really easy for everybody. It's really possible to do, you know, lots of specimens or a few specimens. Even 10 specimens from your yard a year is really, really helpful, because odds are that some of those specimens will be new to science, even from your own yard.
Yeah, and I've found a whole lot of new stuff by doing that, and I'm grateful for the services that you guys, you know, provide to do that. So I do the Myco Blitz every year, and then I started sending OMDL the past few years as well, and it's been very helpful, and it's helped me collect a lot of data, and then it helps us learn what we really do have in Colorado. There's a lot of stuff we didn't even know about, so we're still on that track and still doing it. So this is a really good thing. The one thing that I did want to say is that, you know, sending specimens into this, you really only have to send a little piece. And what I like to do personally is make two, you know, bags per se. So I'll send like a little piece, but I'll keep the main collection here, and have a duplicate. And then that can, you know, eventually go to Denver Botanic Gardens or wherever, so it can be stored in a natural history collection. That way someone can come along later and pick it up and work with it down the road if they need to. So I think for science, that's a good thing. And in the beginning, I was just, you know, collecting and sending the whole bag. And, you know, later on, I'm like, why am I doing that? You know, and then I realized, you know, we only need this teeny tiny piece, you know, to go in a PCR tube. So, I mean, everyone will do it differently, but I think having the whole collection is different than sending a piece off to be DNA sequenced, right?
Yeah, I mean, so James is the top collector in Colorado by far. So he's only been doing it since 2022. So you know, he's
Pretty
your
good
top two.
competition.
Yeah.
There's a girl on the Western Slope who's really pushing it pretty hard, which is awesome.
Yes.
We don't have a lot of stuff from the Western Slope in some of those areas, only in Telluride mostly. I mean, I'm looking at the map there, but I mean, it's really not a competition. It's just, let's get, you know, as much data as we can, so then we can study it and we can learn some stuff.
That's right. But if anyone wants to beat James, I'm not gonna hold him back.
It's absolutely a competition.
Somebody watching is asking if they have dried specimens from years past with iNaturalist observations. Can you accept those?
Yes, we would love to accept those.
Well, so I would say, assuming they're relatively recent, right?
Yeah.
So the older the specimens are, the lower the success rates that we get with our general and cheap sequencing protocol. So we have protocols that we can use for older specimens, but it's more expensive, and we typically don't offer that as the general free service. And so we do try to, we would prefer the specimens to be as recent as possible. But if you have older specimens, we would definitely, I would say, shoot us an email and we can have a discussion on what it might take for those to be successful.
If they're super old, but if they're just from last year or something, I think it's fine.
Yeah, I think if they're like a few years old, right, then they're still pretty much okay. But once you go beyond like 3 or 5 years, then it's
Yeah,
downhill.
you start seeing a big drop off at 5 years with the quick protocol. So yeah, if they're within the last couple of years, that's not a problem at all.
I'm gonna keep going through the presentation really quick just because okay, yeah. There's stuff that, once I get through this, we can like go answer questions and everything. But yeah, so from my last little map, you can tell that there's no baseline for barcoded Colorado macrofungi on iNaturalist. Like we just don't, it's just not there compared to the rest of the United States. So we really need more, we really, really need more collections, especially if you live in a weird place. So like the weird place between Colorado Springs and Kansas, for instance, would be like awesome. That like weird plains area, or like yeah, like you saw like this the northwestern corner of the state doesn't have a lot of collections. So yeah, just really anywhere. There's so many nooks and crannies in the mountains there. So there's places that just stay wet. So yeah, any cool like bogs or places that stay wet. Um, but yeah, so this is just a little how to make a scientific collection. Um, so you can print voucher slips. We don't have like a website for Colorado, so I just pointed you guys to the the Continental Myco Blitz. And is that the voucher slips you want them to use, Steve? I'm just
Yeah, those were fine.
okay, yeah.
So the voucher slips aren't a requirement, but especially when they have the numbers on them, it's often helpful for personal organization. So I would definitely recommend printing off some type of voucher slip and having your own numbering system just for personal organization of specimens.
And then, yeah, PPMS has their voucher slips. Fundus has voucher slips. Really, there's so many places to get voucher slips um, on the internet. But yeah, and it's really cool because it lets you capture this metadata that you might forget about in a couple weeks after you made the collection. And it has a little place for the INAT number right on there. And it also kind of makes it's like a little mat for your mushrooms in the dehydrator so they don't kind of blow all around. And then if you have really, really tiny mushrooms, which we also encourage, I like to use bottle caps for those. So just like a plastic soda bottle cap, flipped upside down. It's really, really good for that. But yeah, so you print your voucher slips, whichever ones you want. Go out and explore diverse habitats. Like I just said, the nooks and crannies of the mountains would be perfect. Or, if you just look on iNaturalist and see where there's no fungal observations, just go there. That would be awesome. Yeah. Take quality photos of each collection. So you want to get the top, the bottom, any distinguishing features, like on this agaricus here, it would be the veil, the base of the stem, the top of the cap. And then you're gonna, for every collection of like what would be assumed to be the same mushrooms, like this collection of agaricus right here, you want to make one single observation on iNaturalist. Once you put these observations on iNaturalist, they'll have an INAT ID number. It'll be different for every INAT observation. And then after that, you can pop them in the dehydrator to fully dehydrate them. Another really great thing is using the dashboard of your car. If you don't have a dehydrator, just sitting your car in the sun on a really hot day, particularly on a Colorado summer day, But also I've really also noticed that mushrooms just tend to just dry out on James's countertop, whether he
Mhm.
dries them or not. So that's really cool. That's a really cool thing about Colorado. So you're not going to be like super worried about that in this particular state. But yeah, we want them to be as dry as possible, extremely dry. Like when you kind of squish them between your fingertips, they kind of crush, they're brittle. They call it cracker dry. So, yeah, once they're extremely dry, you want to put them in Ziploc bags. Everybody kind of has decided that they really like the snack bags. So, like the smaller Ziploc bags that are like this big. And then yeah, with either the voucher slip or just the INET number written on the bag. I prefer to not just write the INET number on the bag. I personally prefer to write it, even if I don't have the voucher slip, on a piece of paper in the bag, just because markers of all sorts, even a Sharpie, can rub off of the bag. And once that's smeared, it's really hard to recover what the number is. And then you're just gonna mail it into Mycota Lab. The address is right here. You can take a picture if you want, or you can just go to the Continental Myco Blitz website, and if you forget, you can just Google Continental Myco Blitz. So it's the same protocol, just a different thing because you guys are open. You're getting the luxury of being open all year in that state.
Mycoblitz.org is a direct link to that now.
Oh, cool. Yeah, okay, so yeah, just mycoblitz.org. And then the best collections will have geotag photos. So making sure your geolocation is on in your phone before you take the photos is a really good thing. Every once in a while, mine will go off randomly. I don't know why. So yeah, just making sure you have the geolocation, because iNaturalist is awesome. It will capture that data and just, put a pin on the map where you were. So there's nothing you really have to do if you're using iNaturalist. It just auto records the location.
One
Um,
thing, can I
yeah.
say something about that?
Mhm.
People collect in different areas and they may not want their location to be, known by everyone. So
Oh,
one of the things
yeah.
you can do is obscure them, which keeps it within a certain radius. I think it's like 20,000 feet or whatever it is. But
Like 10 miles, I think.
yeah, but that will help secure your location. So if you're collecting morels and you don't want anyone to know your morel spot and you want to send something in, then you can obscure it and then it's still valid and still works. So I just wanted to mention that because I know a lot of people are afraid to put the location up of their stuff, depending on what it is. Some stuff's
Yeah.
sensitive than others.
And sometimes even, rare mushrooms, it's good to do that, if you know it's rare. But yeah, I mean, it's not entirely necessary all the time. But yeah, if it's an extra step, but feel free to obscure. It's better to obscure than to make it totally private, because then we can't even tell that it's in the state of Colorado.
Mhm.
Um, so yeah, so if you're gonna be, if you're gonna have these really awesome collections, you're gonna have these geotag photos, all the information on iNet, a fully complete voucher slip in the bag, properly dried mushrooms, which I don't think will be a problem for you guys, and then all the submission instructions followed on the website. And after you read those submission instructions a few times, you kind of just know them by heart, so it's not that much. But we're always here to answer questions. Mandie at mycota.com or Steve at mycota.com. Either one, or info. Um, Here's a cool thing. So this is one of the cool mushrooms that we found in Colorado. This was in 2022. This cool mushroom with a provisional name. Um, okay, NPR came out, recorded Alan, and it actually made it on the local NPR, and then it was picked up by the national NPR. So that was super cool. So actually people do care about this, and we were calling this one the banana russula, but the provisional species name is species TAC 367. TAC is because it was named by Terry Clements, who does a lot of work in Arizona. But yeah, so this is just a really cool one. I don't know if you want to say something about provisional names. You probably will get a mushroom back, a mushroom ID with a provisional name back, and Steve can say a couple words on that, maybe like about what that means.
Yeah, I would say that one thing to keep in mind that most people don't fully appreciate is that most of the mushrooms in across all of North America are still undescribed. On top of that, most of the mushrooms that people think they know in their field guides are actually not the species that the field guide purports it to be. And so when you start looking at DNA of these species and you're able to link things up, all across the continent or between states, we start to realize, based on not just the DNA, but the habitat, the phenology when they fruit, the morphology, that these are actually distinct critters, right? And we can put them into DNA buckets. Eventually, these will end up moving all the species from temporary code names to standard Latin binomials. But at this point in time, we just don't have enough reference data to be able to properly assign Latin names to most of the species, or the species are just new. And so, as an intermediary step, we assign temporary code names to be able to link collections up from these different locations under a single name, just for communication purposes, right? So we can start tracking their ranges, their distributions, looking at these buckets of morphology, even though they don't have a formal scientific name yet. And we're somewhere around 10,000 species in North America right now with these temporary code names. That's outside of the ones that we put a Latin binomial on. And so there's just a massive number of species that are left to either describe or to assign an older name to at this point. And that's going to be an ongoing process probably for the rest of our lives.
Yeah, it would take like thousands of years probably at the rate that we're describing the species to describe all this. So, but yeah, this was a really cool one. And this is around. I mean, it's not uncommon. So if you guys see the banana russula, Alan swears that it smells like bananas. James and me don't smell it, but it certainly looks banana-like.
Fresh, dry. I don't get bananas out of it. I mean, it looks like banana.
Yeah.
I think in older field guides, you might find this under like Russel or Chlorophlava, stuff like that, but which they're related, but they're different. Pretty much any Russel in Colorado is probably going to have a provisional name, with the exception of war.
Yeah, so
just an
Russell is a-
example. I was just going to say for Indiana Russula, we're currently at around 200 species within the state, and the significant majority of those, probably something like 80 to 90%, have temporary code names.
Right.
There's over 50 red Russulas in Indiana. And so, there's certain groups that it's just going to take a very, very long time. And that we might not be able to identify them to species in the field without DNA. And that's a possibility too. But once our main goal right now is linking like to like. So linking these things that we believe to be the same species because of morphology and habitat and DNA barcodes, linking them together in a systematic way. So that way we can start to get accurate descriptions of what the range of morphological variation is, as an example, for these species.
I was just gonna say, so Russell is a really good thing to collect in Colorado. Probably also like Amanita. What else?
Cortinarius, Cynosobe,
Fortnarius, Inosobe. Yeah,
Antoloma.
We have various Ramarias and we don't know what any of them are.
cool. Yeah, and then James happens to be working on agaricus. So if you find any agaricus, he might be able to help you because he has a lot of DNA sequence data of a lot of the agaricus in Colorado now, which didn't exist before. So yeah, just keep your eye out for any of those things. And I think this is the last slide.
Let me give one more anecdote since we're talking about agaricus. Agaricus is one of the genera in Indiana that we believe to have a functionally complete survey of. We don't expect to find any more agaricus in the state. We've been looking and continuing to sequence many every year. And it's been a number of years now since we found a new species. And so that's one of the ways we can track the completeness of our survey is just by the fact of how many specimens are we looking at and how many new species are coming in per specimen. With agaricus, we aren't finding any new species within Indiana. And then beyond that, we have a lot of what's known as environmental DNA. So we've looked at the DNA of soil across the state, and we're not finding any new DNA of new agaricus species in the soil either. And so eventually we'll get to the point to where we're opening up soil collections, as an example, in other locations. And that's going to be a way to help validate our above ground collections, but we're still a few years off from that now. These types of survey efforts, it is totally possible to get to the point to where we believe we have a reasonably comprehensive survey for individual genera, even at this time.
Very cool. I thought we'd never get there in Colorado because we keep finding new ones.
That's really cool. It looks like so I just want to wrap this up and say, please do this. Just do it and get on it. Let us know if you have any questions. And I'm gonna stop sharing, and Brian has his hand up. He's like flagging us down. So yes, Brian. Oh, we can't hear you.
He's on mute.
You're on mute.
Here.
There.
Okay, sorry. I'm just going off and it's muted, which is probably good most of the time. I just got back from the North Texas Mush Fest foray with Amy, Britt, and Carrie, and Sebastian, and David Lewis, who is a Russel expert from the Gulf States area. And we found a few new Russula down there for sure, which was really cool. But Colorado was covered with, and it's not, say the term new, that's not very good because they're not new. They've been around as long as the trees, as their ectomycorrhizal, and even above tree line. We have Chance Knopfsinger found 8 species above tree line with the little dryas, willows, and other little plants up there. We could have, and I was thinking in talking with David Lewis, maybe we could have specific genus forays. Let's say we had a Russula foray, and these are not, this is kind of a new idea to have a genus specific foray. Invite all the Russula people, Tavis, and all of the people, even the Canadians that work on Russula, and David Lewis, and anybody else that wanted to work on Russula, and just hit it. And I'll bet you we could have a really good showing for INAT and for genus Russula, and to make it worth Stephen Russell's efforts there with the genetics. And same thing with Ramaria or anything else, but maybe once a year have a genus specific foray. What do you guys think?
I love it.
Yeah, I would say so. In Indiana, something we still frequently do every year is we kind of look at our data for a given season and say, oh, hey, we need, X, Y, and Z. Here's our lookout list for this year. And ever since we've started, that list has always contained the genera that we just talked about: Russula, Cortinarius, Inocebi, Entoloma, Lepiota leucoagaricus, we often focus on. And so there are a bunch of genera that just need huge numbers of collections still. And I think that would be a great way to kind of focus in people's efforts to get everybody on the same page, to have like a structured and organized process. I would say one more comment for the Myco-blitz each year. We have target genera for the summer and the fall Myco-blitz, across the continent. This year for both the summer and the fall, it's Lepiota and leucoagaricus for the first time. We've never had both of them be the same. Last year it was Hygrocebi for the summer and Hygrophorus for the fall. And so there's definitely those kind of opportunities. We find that to be really beneficial to kind of give people a target, right? To focus in on one particular area, and you'd be guaranteed to get lots of cool and novel things.
Yeah, I started very focused on agaricus, and then once this world opened up to me, I just started collecting everything I saw. And I'm still doing that, you know, and so if I've already sent something in a bunch of times, then I'll usually kind of let it go. But I still like to find stuff from different areas, you know, even though you know what it is already, we like to kind of paint the map, right? So, but specific genus for it would be kind of cool. I just I never thought about it personally, but
Yeah, it works really well in the online format.
very cool.
And for that matter, Fundus has a 20 rare fungi focus that they've got for the Rocky Mountain region right now. I think that's for this entire year, perhaps. Nope, it's the next
That'll
5
be
years.
forever. Yeah,
Yeah.
that'll be like ongoing, but they'll probably pick it up again.
Yeah, I just saw the time frame on the website, and it's the next 5 years. But that's a list of pictures and binomials that we'll be putting out soon, so that people can have that to look for as well. With all of this just being concurrent, does that make sense?
I wouldn't mind sharing a few examples of some of my naturalist records to kind of show some things.
Yeah, go for it.
Let's see here. Share.
And while you're pulling that up, let me make one other point. So even in Indiana, where we have really, really large surveys and really lots of specimens, we still don't have a do not collect list for the state of Indiana. Something that we found is that ignorance is often helpful in terms of these types of collections, because it's very common for people with experience to say, oh, I know what that is. And it ends up being new.
Right.
You know, things like, I don't know, Lactarius indigo in the East. You know, we now know it's like 5 species across North America, but we have no idea what the actual distribution of those species are. And so even the common stuff is good to collect sometimes, you know, up to a point, especially when we're starting out in Colorado and trying to establish that baseline. We want to have representations of all the species that exist. And so that would actually include the common ones for a few years.
Yeah, we're finding that out quite a bit. Like, you know, Lacaria Licata, we've always called it because the books call it that, and then we're finding it's something slightly different most of the time. So here's my iNaturalist. Like here's Crassosporium I collected recently. And so what I like to do is take a lot of photos. When I first started, I took like 1 or 2 photos, but what you'll find is a lot of people just take a picture of the cap and then they don't have anything else, and so it's hard to identify it. And I go through iNaturalist trying to identify stuff quite often. And, you know, when you only have the cap of an agaricus, you know, it's an agaricus probably, but you don't know any much more than that. So like Mandie was saying before, it's good to have pictures of the gills, the stipe, the whole fruiting body. I like to have, you know, pictures of the mushrooms actually as they're growing, but then take a few out like this. And I actually learned a lot of this from Alan. He's great at the photography, and just organizing the mushrooms, you know, is important, I think. You can also take it a step further if you do microscopy. You could include pictures of spores as well, or any other microscopic features like basidia, like right here. Not everyone is going to do that. I also take pictures on a separate camera, which I might bring in later, and then include things like spore measurements. So I think the higher quality your iNaturalist observation, the better that data is for the future. You know, rather than just taking one picture, try to take several. Try to obviously get the top, the bottom. Cross sections are good as well, depending on what you're collecting. This Colibia is upside down here, so you can see the bottom of it as well. And then I go through here and look at stuff. Here's all the ones with DNA sequence data on iNaturalist. So one of the things I like to do is verify that these are legitimate. And I know that you know, with MycoMap it's a little different. I actually need to talk to you guys about how to use that properly. But what I do is I go through, and here's a new agaricus that was posted by Jonathan Frank, and this is in the southwestern corner of the state. And so this is actually one that we didn't know existed here. I did learn about this recently from a mushroom observer observation, but then he just posted this up here last month. So what I do is I'll see his barcodes right here. So you can take his DNA sequence, copy that. And then what I like to do is go over to, run a blast. I know that you guys do it a little differently. If you do it in Google, you can type in like NCBI blast. But this will give you some basic information. So I go in here, I paste the sequence in here. So this is Jonathan Frank's sequence. I pasted this earlier and I was curious. I'm like, is that really what it is? And then you do a blast and it's going to give you some results, hopefully. I like to sort it by, the highest percentage ID here. And so you see that there's, here's a fundus one, matches 100%. And then here's a few more, here's a couple more. And so it looks like it's pretty close to that. One of the things that I also like to do when I do that is I go to alignments and I switch this over to dots with identities. That way you can see what's different. So this one's 100%, so there's nothing really different. Then you can go to the next one and so on. And sometimes they are different. These are all 100%. Here's one 99. So you see that this one letter's different. So it's got one position different. I just find it's very easy to sort that out. If you select that from the pull down, you can see the differences easily, or gaps like right here. So that's what I do. And then I also go, okay, well, how legitimate is this one? So I'll click on this one that matches 100% from fundus. And I say, okay, who did it? And then you start looking down, who named it this? And you see this was authors were Scott Ostoony. Oh, hey, this looks like a lot of the people in OMDL to me, but and then you can see who the collector was. So here's the collector, and you can see where it was. So that's in California. So basically, the Agaricus Bruno fibrillosus that Jonathan Frank posted matches 100% with ones from California. So what I did is I went to his iNaturalist record over here and I agreed with it because I agree that that's correct. And I did that right before we started talking. So I oftentimes go through and try to verify on my own. I know there are other tools to do this with, but this is a really easy way for other people to do it. I just wanted to show that. When I get sequences back from either OMDL or from Mycota Lab, I often go and I look it up and I try to verify that if Steve calls it something, I try to verify that that's correct. And if it's correct, I agree with it. If it's not, I may dispute it. I may say something, depending on what the criteria is. This observation is good because it does have a DNA sequence, but if you notice, he's got one picture of it. And so I think it would be preferable to have more than just the one picture. I like to see the top of the cap and see if it's, got some color to it. Just, other features. And then again, with Agaricus, cross sections, I think are important. And I often forget to do that. So I'm on mine here. I'm gonna pull up all the Agaricus I have in here.
Go to one that was OMDL or Mycota, because then you can just,
This one recently came back right
there's
here.
the Mycomat blast link, and that's much easier. You don't have to go to NCBI. You can just like click that. Yeah, click
Right
that.
here.
So that's how people can really see. If you just
Mhm.
click that link, it's like so much easier. It cuts through all that, like, oh my God, what is this other website thing? And it just takes you directly to the screen.
Right. And I think part of that is also learning how to use Mycomap and understanding what this information is, which there's a lot in here.
Yeah, that's
Here's the ones that that's referencing, which is right. Agaricus argenteus, that's it was an illegitimate name. It's actually Brandelize, the real name. And so, obviously this is matching those, matches one right here from Steve
yeah.
Wilson.
But no, people, I mean, if anyone wants to know about this stuff, you can obviously ask James anytime because he'd be super happy. But, you don't have to do this stuff. It's like totally not required.
No.
Someone
If
will
any
put
of
their
this just
ID.
went above and beyond anybody, or if this just suddenly sent anybody into overwhelm mode of like, that's too much for me, that's okay.
Yeah,
This is
and it
not for
could.
you yet. Everybody can work at the level they're at. If your level is collecting specimens and taking pictures and uploading it, great. And
Yeah.
then there will be people who can take things a step further, or if you're already at that level of the photography and the posting, then You know, that's up to you if you want to step up your game and learn the microscopy aspect or start studying and analyzing the DNA samples. But none of this level is required for this project.
It's not.
It's no, it's not. It's cool for people that want to do it. I just want to make sure that this is not tipping people into overwhelm, that they're going to back off and say, whoa, that's beyond me. Yeah, it's beyond me.
Yeah.
But I might practice that this year.
Yeah. And that, like I said, James is a great person, or me or Steven or Alan or whoever. But me and Alan will be there on August 8th and 9th. So we will be in Colorado Springs those days. I'm going to give a talk and Alan's going to give a walk. We've already planned it. So if you guys have any questions, if you want to learn more about mushroom photography or more about this stuff, we would just sit there and, you know, blast stuff for people if you really want to see it. But if not, you know, it's just whatever, however we can support you in any way. Like I feel like you guys are so lucky, honestly. I wish someone would have like 10 years ago been like, yeah, let me like help you with like all this stuff. And like, we're just really happy to be at this level and at this point and being able to support anyone in Colorado that really wants to try to even just start get into this. And then if you are more advanced, I also want to say if you really, really resonated with what James just showed, and you want to learn more about that, there is the Volunteer Sequence Validation Committee. It's called the VSVC. You can, I think, just Google it. I think it's also on the Fundus website. They are going to have a new cohort for that, so they're going to be training sequence validators. And with the stuff getting sequenced by Mycota Lab, you can pretty much validate your own sequences. So if you really are interested in that, if you've been doing this for a while, you know,
I think most of the time, a lot of people will just send stuff in and they won't take it to this level.
no.
The NCBI has a lot of good data, but Mycomap has even more data than that. So that's where this is powerful, I think, and learning how to use it can, take a little time. So you really don't have to use this stuff, and someone will come along. Like if you have, something with a DNA sequence, sooner or later, I'm probably going to find it and I'm going to go and try to validate it and make sure that, what these guys are saying, is correct. And that's just double checking work and making sure that that's really what it is. And most of the time they're right, because they have all the tools and they know what they're doing. And they even have more tools than I do or someone else would. But I just wanted to show that in your record, I mean, obviously you're going to get this ITS barcode right here, and you'll be able to take and do stuff with it if you prefer to. If not, some smart person is going to come along and look at it and try to contribute. There are a lot of people who go through iNaturalist records just to help identify them. And this data is really important because this research grade stuff will be exported later by other scientists for fungal conservation. So I'm actually part of a group that's doing that. And, the more valid this data is, the better for the future for developing red lists and rare list species. So, and
yeah,
then
I would just reemphasize that point. If all you want to do is take pictures of mushrooms and dry them out and mail it in, that's all you have to do. Somebody will come along and identify it for you. We'll do all the DNA analysis. You don't have to worry about that, but you can get as involved with it as you want. The options are there to keep on expanding your knowledge in the subject area.
Right. I just got hooked on going through and trying to validate things myself. And I just really want to know. And I love the information. So I'm grateful that we have these kind of services, and I think more people should use them. There shouldn't be, a handful of people in our state doing it. I think anyone in our state who likes to go out and, hunt mushrooms and want to contribute to community science, this is their way to do it very easily without spending any money, really, So
Yeah, I'd say almost everybody that is a power collector, who ends up going out and making lots of collections within the first 2 years, they end up starting to learn about the DNA analysis process, just so they can help so they can learn more about their own collections and their own results. But especially starting out, you have to know nothing about DNA, and you don't even have to worry about it. But if you want to, over time, you can start to pick up that skill set.
Right.
You'll naturally become a better mushroom identifier too. So even if you just use it for that, just having these, accurate and reliable IDs,
Yeah, and it's made me a much better mushroom identifier. I've learned so much by doing this stuff, and you just have better information, a lot of the stuff that we reference is outdated. So here's one that's really cool. And I have Alan and Steve saying it's Olympiana, which is what I believed it was, but they still haven't posted the barcode. So I have a hard time validating this one, but I'm really happy that this came back. This is the one that came back recently. So this is what we were calling Russel zerampelina, and that's a European name. So we have that group in Colorado, but this is was recently named a few years ago, and I believe it was by Chance Knopfsinger. There's a paper on it. So we have several in that group. So what I'm trying to do is collect all the ones from that group and try to find them all. That way we can actually put the names to them. So I haven't agreed down here yet. And the reason why, we know it's in this section, it's probably these guys are right. So Alan and Steve say that's what it is. I'm assuming, because of the DNA sequence, but there's no data yet. So what I do is I just wait and hope that it shows up.
I dropped a link in the chat to where you can search by iNaturalist number and find it quickly.
Oh, perfect. Thank you. Yeah, I know there's another way to do it. I need to start using the tools you guys have because it would be helpful. Oh, there we go. Thank you,
I'm
Steve.
saving all of these links from the chat so that I can share them to socials later for people.
Yeah, and I just wanted to put a visual thing up on the screen and say, hey, here's kind of what we do, and here's how some of this stuff works, and that way people can visualize it. When you're using iNat on your phone, it's a little more limited, but the great thing is that you take a few pictures, you just share it right to iNaturalist, and then you upload it, and then you walk away, and you just keep doing that, and then you have a number for reference, and then you can, do vouchers. If anyone needs vouchers in our club, I have a Word doc of these. I've been using the one that Alan's been asking people to use. He put it out there in the public, which is really cool. This is an example of one from your son, Meg.
I can
But
send you that, Meg.
it has the QR code, which will link right to the iNaturalist observation. It speeds up the process. I use that quite a bit just because I can just put in a bunch of numbers and then print out a page, and then I don't have to write it down. And then we know the numbers are correct because I check them all and just copy and paste the iNat number. But
Yeah, Alan has it,
Brian,
but it'll
can I
flag if
talk?
it's not correct.
Yeah, I just have one question. So we send stuff to Stephen Russell, of course, and it's wonderful. Is that only ITS or is LSU DNA being considered also?
For now, it's only ITS, so we're kind of rethinking that. It's probably going to stay only ITS. For 95+% of species, ITS alone is going to be wholly sufficient for an identification. There are a much smaller number of what I would even call edge cases to where it's not particularly helpful. There are certain genera where it's less helpful than others. But for an initial pass, the first pass is just going to be ITS. We're currently considering what to do for a second pass for very targeted groups. So anything that's unclear with ITS will eventually have another gene region done, but not initially. And we're still considering how we want to structure that because we're going to have to go back through a whole bunch of targeted specimens. But it would be done in a targeted way. It wouldn't be for everything.
Okay, and the other question is, as we dry mushrooms, we have a lot of different ways, whether it's the dashboard of our car or what, but is there a recommended temperature in Fahrenheit for drying mushrooms for DNA?
Dehydrator under 140.
Okay, good. That's excellent. I know I've cooked a few on my dashboard, and I cooked an unusual bolete. And when I gave it to Jonathan Frank, he couldn't get anything out of it, even though it was a fresh specimen only a few days before. But going to Arizona and then having the windows rolled up, that cooked it.
that'll do it. Do you guys, what What do you guys think your success rate is on stuff that's sent in?
Yeah, it really depends on the collector. I would say that's kind of the biggest X factor. So, I mean, in a general sense, we put everything through anything that doesn't work the first time, we put it through the second time. One, once it goes through 2 passes, it's probably at like 95% success or something like that. I mean, for the first pass, maybe, yeah, it depends, maybe 80% or something like that. You know, I'd say 70 to 80%, depending on the collector. And we pick up another 25%.
And then, so let's say we start sending stuff in by like, I don't know, September. What I typically do is I collect all year long and then I send a big batch all at once generally. Sometimes I send two, but it takes me a while to process everything from my end and then I end up mailing it off and it takes a little time. But how long? I'm asking these questions for the viewers because I kind of have an idea, but like how long after sending, you know, do you think people will start to get data back? And then if they don't get data back, you know, will they be notified that it failed?
Yeah, so, and that's actually a more difficult question as we're scaling up, the timeframes shift. Last year, our lab processed something like probably on the order of 30,000 specimens. And so it's, you know, the numbers continue to climb and not go down. Typically, we would have data ready within 2 to 3 months, but then that's not fully analyzed. That's our biggest bottleneck right now is analysis. So even as with the one we just looked at, the agaricus, me and Alan had both commented on it, but there wasn't a DNA barcode pushed to it yet. And so the analysis is taking a little longer. We're currently working on a system to automate most of the analytical functions. And so that's going to hopefully help out with that, the current bottleneck for analysis.
Okay.
We talked about the volunteer sequence validator core as well. For people that know how to validate their own stuff, it's possible to create their own projects and to have it all on the dashboard as soon as the data is ready. And that typically doesn't take more than 2 to 3 months.
Okay. Yeah, I'd definitely be interested in that in the future. Right now, I'm a little have too many things on my plate to be a validator for everybody else. I'd love to do that though.
One other thing I wanted to ask, and I may have missed it. Do we have your address, Stephen, where we send these specimens to? I know we're out of time here tonight, but I just want to make sure that you post the address we send specimens to. I may have missed it.
Yeah, it'll be on the links that'll be shared.
It's
Wonderful.
on the Continental Myco Blitz. It's on the bottom of the Continental Myco Blitz website.
Okay, wonderful, great. Wow, great job, you two.
Yeah,
I've got a question from Instagram. I don't know if he's on watching or not, but he's been trying to do his own PCR at home and has hit significant barriers, including access to primers and other lab chemicals as a citizen scientist, and would love to address those. Do you guys have any suggestion or support for him?
Yeah, I would say there's a fungal sequencing group on Facebook that's dedicated specifically to those kind of questions. And
Okay.
so there's two main Facebook groups, fungal sequencing and fungal sequencing results. And so, you know, if you have a question on just the process and you're trying to do it yourself, there's no better resource probably globally for that. And then there's a fungal sequencing results group where if you're having issues interpreting the data that you're given, you can post on there and people will help you out.
Okay, awesome. Thank you.
Yeah, thanks for watching, guys. I'm really happy that it looks like we have 16 people on there right now. So that's really, really cool. And, you know, hope more people end up seeing it. And
Are we getting any questions on YouTube?
I think we've answered all the questions on YouTube. This will be available forever on our YouTube channel, which is the Pikes Peak Mycological Society page. I will share the link on Facebook. Meg will share it on Instagram, and so that everybody will have access to it who didn't get to watch it, who would like to watch it again. We do appreciate you guys coming. Thank you so much for that really fascinating talk and information.
Yeah, let me make one more point to close if I could.
Sure,
What
please
we're
do.
really, what we're really trying to do here is build foundational data sets. And so there's a lot of researchers that would like to ask a lot of questions about all of these species, but there's very few species where we know the true range, the true distribution, the true phenology, the true morphological variation of these species. And by starting to organize these collections, by making these physical collections, and by doing DNA barcoding en masse, it's going to form the foundation that's going to allow future generations of researchers to be able to ask questions that they can't ask today because the data doesn't exist. And so, by starting this in Colorado and by expanding it and by, working to get thousands of specimens into this kind of process, you're going to be helping generations of researchers be able to ask questions that they just simply can't ask today.
That's
Very cool.
Thank you.
Good stuff.
Very cool stuff. Thank you, guys. Thanks a ton. And you're always welcome in Colorado, right?
Absolutely.
Yeah, I'll be in Colorado, like I said, I think it's the 8th and the 9th of August, and then I'm going to be at Telluride, and then I'm going to come back to Colorado, and I'm going to do a presentation around Denver, and then I'm going to go up to Longmont and do a presentation. So I have like 4 presentations in Colorado. So if you want to catch me, I'll pretty much be in Colorado for like 2 or 3 weeks in August. And you can just flag me down and I'll help you with anything, and we can go into the woods and it'll be really fun.
Yeah, well, we'll see you at Telluride for sure.
Thank you.
Sure.
Okay.
Yep.
Bye, everyone.